Naimit, the project, has 6 work packages that deal with content. In each of these work packages, we focus on a part of the immune system that we try to modulate with natural immune modulators like vitamin D, for instance, in such a way that the immune system becomes now tolerant to the insulin-producing beta cell. In other work packages, we rather focus on the beta cell and try to understand how the beta cell reacts towards the attack of the immune system. And if we can understand that, we can also find ways to make stronger beta cells.
So currently, Tina is plating a lot of bacteria. Actually, she's making a lawn of bacteria, which is a quite important step in the process of getting a bacterium genetically modified. The result of this plating will generate plates which look like this, where we have used such a protocol that the non-modified bacteria turn blue, whereas the genetically modified bacteria will become white.
In work package 4, we exploit the gut as a way to introduce new antigens towards the immune system so that the immune system will become tolerant towards these antigens rather than become immunogenic against these antigens.
Antibiotics are live Lactococcus lactis engineered to secrete and to deliver therapeutic proteins directly to the gut. Our participation in Naimit, in the Naimit program, involved the engineering of such an antibiotic that delivers an autoantigen, a proinsulin, along with an immune modulating or tolerance enhancing factor like interleukin 10.
Now the focus shifts from purely looking at the immune system towards looking at how the beta cell reacts to the immune system attack and how the dialogue between beta cells and the immune system comes about.
Up to today, most of the research in the field focus on the role of the immune cells, and the pancreatic beta cells, the target of this autoimmune response, are often seen as a passive, a victim of this attack. And what we and other colleagues have discovered is that this is not a completely unilateral phenomenon, that pancreatic beta cells actually keep a dialogue with the immune system.
I am presently extracting some mRNA from pancreatic beta cells to see the transcription and the regulation of the genes. By this machine, we quantify the copies of cDNA that we have in our samples. Once the run is finished, we compare our samples with this standard curve, and we obtain the values of the mRNA that we wish to quantify.
And our job in the Naimit project is to understand which are the signals that pancreatic beta cells trigger that will lead to their death or to their survival, and try to modulate the signals in such a way that they will be less perceived by the immune system and hopefully will be protected against cell death.
In work package 1, we study the dendritic cells, the cells that are crucial, central in the immune system in presenting antigens towards other cells of the immune system.
So here we are culturing the immune cells of the patients because we want to use them as a vaccine to treat them with vitamin D and inject them back into the patients to help the immune system of the patients fight the destruction of the beta cells in the pancreas.
We use natural components to change the immune system, in our case vitamin D3. Our contribution is part of work package 1, which is also the final stage of the pipeline from bench to bedside. So we really try in patients to change the course of the disease by protecting the insulin-producing islets of Langerhans with a cell therapy.